Hello everyone, my name is David Crossman. I am the Director of Bioinformatics for the Heflin Center for Genomic Sciences and Associate Professor at the UAB Department of Genetics. I am here today to talk to you about whole genome sequencing analysis. This can also be considered analyzing exomes if you don't have whole genomes, even doing a targeted approach. I'm going to walk you through the steps of how to analyze this data to call variants in particular patients or samples or cases of whatever you are trying to call variants for. I'm going to run through the Broad's GATK pipeline. So we're going to stay in one of the version 3.6 or 3.5. Not moving to version 4 yet. That just came out about a week or so ago, and I haven't had a chance to really play with it. This doesn't discuss anything about the Google's deep variant tool as well. Those, along with the new GATK version, changes a lot of things around. So it'll be interesting in the coming months for me to figure out what that is doing. All right, so say you are given a case of a mom and a dad walked into the genetics clinic and their child was sick, and they'd seen doctors all over the nation, and the doctors were not sure what was going on. And so they decided to get their child, along with the mom and dad, exome sequenced. And here you're given the FASTQ files. These are the raw sequence files that came from the Illumina sequencer. So what do you do with those? So we're going to run through this tool set. On my right-hand side, you'll see the steps that we are going to follow here in the GATK practices. So you can consider it three different phases: pre-processing, variant discovery, and call set refinement. So you begin with the raw reads. We step outside of GATK for the first couple of steps, so we will want to map to the reference genome using BWA. We will then go in and remove duplicates, and then we come back into GATK to do some base recalibration, and at that end of that phase, we now have a analysis ready type alignment file, a BAM file, B-A-M file, that you can then take into phase 2 to do variant discovery. And so here we'll use the haplotype caller to call variants. We will combine them to get our raw variants, so we'll have both SNPs and indels. We'll filter them, and at the end of that we'll have an analysis ready set, which then you move into phase 3 and to do a call set refinement. And here is a lot of tools that I won't be able to cover in great detail because there's a lot of variability here, a lot of different ways you can go about analyzing this data to refine your genotypes, to annotate them. And then at the end of the day, you need to evaluate it. And if it looks great, you're great, move on. If not, you need to go back and troubleshoot some steps here. So before you can even start any of those steps, you need to find the reference genome that you're going to be aligning these raw FASTQ reads to. And so we're using the human genome. This was the mom, dad, and their child. And so we need to find the human genome, and I'm going to pick HG19. And in this instance, this example that we're going to run through today is only looking at chromosome 20. And so I extracted the chromosome 20 from UCSC, and we need to index it with BWA. And note that you only have to do this once. Once you index the genome, you're set. You don't have to come back and index it again. So a lot of different DNA aligners are out there. GATK recommends BWA as the aligner, and so we're going to go and use BWA. You'll notice here is my terminal, and down below is just the the directory structure that can be visible for those that aren't that familiar or are still scared of that blinking cursor up there in the terminal. And so we need to go into the genome directory. So I'm going to change directory into genome here in a second, but I wanted to show you what's inside the genome folder. And so here you'll notice that you have the FASTA file, which is contains chromosome 20 from the UCSC-HG19. So let's go to terminal, and I'm going to change directory into the genome folder. And so I'm now inside the genome folder. If I were to list the files there, you'll see the same three files, the DICT, the FA, and the FAI. They match those that are down here in this directory. So first, let's index the FASTA file. So we want to take this line of code here, and we can copy and paste it. I'm going to type it out just for the first couple of chimes so that you can see that typing isn't bad. So we will index. So basically the command is BWA index, and then you provide the FASTA file. And you'll notice that there will be new files that are going to be generated here that are being created from the BWA step. Okay, that took a couple of minutes to run, and that is now done. And when it's finished, you're brought back to your command prompt. You'll notice that it ran through various different iterations. A whole set of files, new set of files were created here. So if we were to list the here we go, list the files, you'll now see instead of the 3 files we had, we now have about 5 or 6 more. And so it creates 5 files with the extensions, these various extensions here. All right, great. You have your genome now indexed. If you were indexing the entire human genome, this would take several hours. Doing just this chromosome 20 took a couple of minutes. So prepare to just sit back and let this step run when you first set that up. All right, so the genome has now been indexed. We are now ready to start processing our raw sequence files that we got from the sequencing center. We want to find out what is the variant or variants that are causing the child to be sick. And so first step, even before we even start alignments, is that we want to do some sort of quality control. And so we typically here run FastQC, which is a very fast and powerful set of tools that just does quality control metrics on the raw FASTQ files to tell you whether or not you might need to do some sort of trimming or you might have a bad set of reads that need to be removed. So let's get to that. So we're currently in the genome directory, and we need to move over to the whole genome directory. So wherever you put your files, these steps can be independent of where I have them, but you just need to be aware that where you have your FASTQ files and such. Here under whole genome, I wanted to show you that I have 2 sets of FASTQ files for each individual here. So I have the father, I have the mother, and I have the son. And I have read 1 and read 2. And so this is paired in sequencing. All right, so let's go change directory. So we're going to go back and go into the whole genome directory. If I were to list the files, you'll see the same files there that are in the directory structure below. I am going to go ahead and make a FastQC folder just to store things. I like to be very well organized, so I like to create folders to put things in so that I can find things a little bit faster. You don't necessarily have to make this folder if you don't want to. It just makes things easier to find. So I'm going to create a FastQC folder along with a subfolder called trimmed, and you'll see now that I have a folder created here. If I were to list the files, you'll now see the FastQC folder is there. All right, so to run FastQC, we are going to take this line of code here, and you can copy and paste that line, or we will type it out. So FastQC, here are several parameters that I am going to use. I highly suggest and highly recommend that you look at every tool that you're running and understand which options, which parameters you need to use to call your tool to run the analysis. So my no extract, no group is just going to basically not extract any of the files. It'll keep them gzipped and so forth. All right, so, and I'm putting it into the output folder FastQC. and I'm going to use the wildcard, the asterisk, to call fastqc to run on every single file that ends with the .fastq.gz. Once you have that typed, you hit enter, and it will run through each file, and we will look at the results here shortly. All right, so that's run. Again, this ran pretty quickly, a couple of minutes, not even that, to run through those 6 FASTQ files, 2 for each of the family members. This is because this is only of a very small region in chromosome 20 that I'm analyzing against. If you are looking at the whole genome or exome again, this would take a little bit longer to run the FastQC. All right, so let's take a look at the folder that was created here. So if you go inside FastQC, you'll see that you have the zip folder of all the results. You can unzip this and look at all the various text files that were created. But here I'm going to show you just the HTML file, which is a nice visual to see what has changed, or not really necessarily what has changed, but what could be problems that you might need to address before you can start the alignments to the human genome. All right, so I'm going to just maximize the screen here so we can see this better. Here on the left-hand side is a summary of all the various metrics. You'll see nicely that they have green check marks, red X's, and yellow exclamation points, sort of to point you in the direction of what you might need to look at. This is created for every single FASTQ file, and so I'm looking at just this particular FASTQ file right here, this father read one. So here you'll see just a basic stats. So there's about almost 27,000 reads, sequences here in this particular FASTQ file. The length of the reads is 151. So this is a paired in 150 experiment. GC content is 39. That just gives you some basic stats. Now, again, this is a very small excerpt of this particular genome, and we're only looking at a very small region of chromosome 20, and so you would hope to see several tens to 20, 30 million reads here, and your sequence length is dependent upon how long you sequence them at the sequencing center. If we go down to per base sequence quality, this gives you an idea of the quality score, the Q score, the Q in the FASTQ file, of how well the sequencer understood that the base at that position was an A, C, G, or a T. So scores 30 or greater, so 30 to 40 is your best score. 29 down to 20 is, yeah, they're okay. You might need to mess with, might need to modify them. And anything below 20 is something you need to sort of look at to consider what needs to be done. So with this, we'll scroll across, and we see that the quality there is pretty good all the way down to about the end here, probably about the last 3 to 4 bases, maybe even the 5th base. The quality starts to drop, but that's common inside the Illumina technology, that as the sequences go further out, the quality starts to drop. And so for that, you could come in here and trim those, remove those last 4 bases if you feel that that's causing a problem in your downstream alignment. For this example case, I'm not going to worry too much at the moment about that. So let's keep going down here to look at some of these other FASTQC metrics. Per tile sequence quality, it had a red X. I typically am not too concerned with this type of tile format, but you'll notice that the last couple of bases, probably about the last 20 or so bases at these tiles, they were bad. That could also lead to a possibility of why these might be dropping off as well at the end. Per sequence quality score is just a very simple line chart of just showing what was the most, or do you have a different set of spikes that would tell you what your quality is. You can see most of the quality is really good, sitting here around the 28. You don't see anything here that would be a spike at like 9 or 10, which would cause some concern that you might need to do some sort of trimming or removal of reads that are bad. Per base sequence content, depending upon your genome, if it's a GC-rich genome, you should see the GCs higher, but you also want to see sort of a level set of lines going all the way across for all 4 bases, which you do. Per sequence GC content, the theoretical versus the actual read. Here's another one that I like to look at, this per base in content. Is there any point in the sequence reads where the sequencer did not know whether it was an ACG or a T, so it assigned it an N? And typically this occurs if there's bubbles in the flow cell lane where the camera cannot focus through the bubbles, and so you'll see a spike somewhere, one of the base reads that would indicate that you have a bunch of N's. Sequence length distribution, this is more of a useful chart. If you're doing trimming, it'll tell you the lengths of all your reads, but as you can see here, all the reads were 151 bases in length. Duplication levels. For exome genome sequencing, you do not want to see a large spike over here at the end, on the farther right of the x-axis, because that would indicate there's a lot of PCR duplication over amplification artifacts that you'll need to remove. Here, this looks pretty good. Overrepresented sequences. Right now it's showing nothing, but you might see a primer adapter or something else that just seemed to have been sequenced over and over and over again. Adapter content, this gives you an idea of whether or not you have any of your primer adapters, and you'll see the 5 that they look at. You can add more if you need to, but here it looks fairly good. There's a little slight uptick there. But overall that looks okay. And then K-mer content for those that are that focus in on the K-mer content. This will give you an idea of where those are at. So overall, this file looks good. I would encourage you to look at every other FastQC file report to see what needs to be done, what needs to be modified. So let me close this. And for this example, I'm not going to look at all the rest of them. I know what they look like, but I wanted to sort of explain that in my documentation, I give the various parameters options that I'm using, but there are a whole wealth of other ones as well to look at. So make sure you read the manual for those particular tools. All right, so I really didn't see anything wrong that was too drastically wrong with any of those FastQC reports, but I wanted to show you how to trim the samples. And so we're just going to do a very basic, simplistic trimming of removing the primer adapters from the sequence reads. And so I like to use a tool called Trim Galore, but there are plenty other tools out there that you can use, Trim-O-Matic, Cut Adapt, which this Trim Galore uses Cut Adapt. So be conscientious of what each tool you want to use. They all do roughly the same thing. So just make sure you understand the tool and use the options and parameters appropriately. So for this, we need to trim each of the samples. So we need to trim the father, we need to trim the mother, and we need to trim the son. And so here we're using Trim Galore. We're calling this is paired in sequencing, and then I'm going to have it run FastQC on top of this on the trimmed read, so that way it will know. That way I can come back and look at it, and then I'm going to output it into that trimmed subfolder in FastQC, and then I provide the forward read and the reverse read. So let me copy this and see if the copy paste features work here. Looks like it did. And so you saw me just copying and pasting, but you would at the beginning need to type that out. So let's see if that runs. Yes. Okay, that took about a minute to run, just the father. We need to go in and now do trimming on the mother's samples. And so I'm going to hit the up arrow key to bring back the last command I typed at command line, and I'm going to erase the 2 FastQ files that were linked to the father, because I want to keep the same parameters of Trim Galore the same across all 3. Yes, I could copy and paste the mother's command if I wanted to, but I wanted to show you that I can just come in here quickly and start to type in mother. I hit tab to autocomplete, and I added R1 so that it knows it's the read 1 file. I'm going to go in and do mother R2, and then I hit enter or return, and this will start to run trim galore on the mother's samples. Okay, so the mother's samples have now been trimmed. We're back at the command prompt. We now need to run this on the son. So I'm going to use the up arrow key again to bring back up the last command. I'm going to erase the two FASTQ files of the mother, and I'm now going to put in the files for the son. Again, these are going to be based on what your name of your samples are. So replace the son R1 with whatever your read one is for whatever the name of your sample. And then for R2, do the same thing. All right, we'll hit enter, and we'll wait a minute or two for this to finish. All right, that finished. So we now have our trimmed FASTQ files. And let me show you where those are at. So I'm inside the FASTQ folder. I'm going to back up one directory into the whole genome. And so when trim galore finishes, it's going to create these trimmed FASTQ files. And typically trim galore likes to end the files with an fq.gz. And then several other additions to the file name. So these are your trimmed files versus this was the original file that we trimmed, or that was given to us from the sequencing facility. Okay, and that there you should see one for each of the samples that you've processed. Now, me being a neat freak who likes to keep things clean, the trimmed FASTQC reports went inside the FASTQC folder, and there's a trimmed folder. And so here you see the same type of files that you just saw earlier in the FASTQC, but these are of the trimmed report. So if we were to open up one of these, this will bring you out to the trimmed of the father read one. Here you can come in and look that we have about 25,756. I'm drawing a blank on how many we had before, and now the sequence lengths range anywhere from 20 to 151. I'm not going to spend too much time here looking at things, but the quality scores are a little better. Sequence content went worse, but that can be fixed by setting some other options and parameters inside trim galore. Still no in content. My sequence length distribution is still roughly somewhere in the 150 range, but there are some shorter reads there. So I'm not going to go into the rest of this information, but I encourage you to look at this to make sure that the what your FASTQ file is that has been trimmed or modified in any way, that it looks good enough for downstream alignments and processing through the GATK pipeline. One other thing about you'll notice inside this whole genome folder is that there is a trimming report text file that is created, and it basically is a snapshot of everything that was flying across the screen as your data was being analyzed. And so opening up one of these files, you can sort of see which primer adapter it found, how many reads actually had the primer adapter, and so forth. So this gives you a good sense of how contaminated there were of primer adapters. We only looked at primer adapters, but you can also come in and say remove the last so many bases from the 3' end, or remove the bases from the 5' end, things like that. So look at the trim galore manual to understand what you can and cannot do with that tool set. All right, so I like to just clean this up a little bit. I don't like to have all these report text files sitting around, so I'm going to make a directory, and I'm basically going to follow these two lines here. I'm just cleaning up my directory. So I'm going to make a directory called trimming report, and then I'm just going to I'm going to move everything that ended with report.txt into the trimming report folder, just to clean up. And also for me to easily find the files of the trimming report there. Okay, so here I've got my options, explanations of the options I've used, sort of an explanation of what happened and what we saw. All right, great. So we've now completed the very beginnings of the analysis, before we even started the GATK pipeline, you need to do this for every single sample to make sure that the sequencing facility that sequenced your samples, that there's nothing wrong with them. So FastQC, find out the quality control metrics that, and if there needs to be any sort of modifications done, then you use some sort of trimming type tool. And then after trimming is done, you want to rerun FastQC and to make sure things look good. And if things look great, then you're ready to start your downstream analysis. All right, so I'm going to say that I'm ready. I don't need to do any more other trimming or anything else. And so now we're going to start into the GATK pipeline. And we're going to start with the first phase, the pre-processing phase. And so here we're starting here at the beginning with the raw reads. And we're going to map to the reference genome using BWA-MEM. Remember, at the very beginning of this, we indexed the human genome of only chromosome 20, but you would index it whatever genome you're using. We will have 3 commands, one for each of the patients here, the father, the mother, and the son. So this is the command that we would run for the father. This is for the mother, and this is for the son. And I'll walk you through each of these here in a second. So we need to copy and paste, or if you felt like typing all of that, good luck. Here we go. So I'm going to hit enter after I copy pasted that. And that's going to start the alignments. So let me explain the what I ran, and you can see it ran pretty quickly because it was a very, very small region of chromosome 20. This step is also a very time-consuming step. If you had a whole genome, this can take several hours to even days, depending on your computing cluster or whatever computer you're using to run this type of analysis. So here we're using the BWA command. We're using the MEM aligner part of BWA. Here is the M-M option or parameter. It's just going to mark your shorter split hits as secondary. Helps with Picard. The -R is for read groups. It's important in GATK that you include read groups. So here you provide like the ID, the flow cell ID of the Illumina flow cell. Here I just labeled it ABC123. You want to provide some sort of library name. So I was giving this the father's name. The platform is the Illumina, and then just the sample name is father's. A lot of this can be modified to however you want this to look at. I encourage you to read the GATK best practices guidelines to understand this a little bit better. All right, and then once I give it the read group, I then will go to the directory where I have my FASTA file. So remember, we were in the genome directory, and I point it to the FASTA file that I need it to align to. And then I will then provide the read 1 of the trimmed FASTQ file, and then read 2 of the trimmed FASTQ file. I then will send all the information out to a SAM file. This is your alignment file, so I'm going to label it father_aligned.sam. And so that will create a SAM file, a text-like file. So let's go on and repeat this step with the mother. I'm just going to copy and paste because it's a lot easier at this step, because there's a lot of modifications if you were to go back and just use the up arrow key to change. And I would be prone to making mistakes here. So that one finished, and these run fairly quickly again with a very, very small amount of reads. Don't expect this to be the case when you're looking at whole genome data. Here I'm going to run the sons command, and I'm going to hit enter, and that finished. So let's look at what these SAM files look like. So here you'll see you have this father-aligned SAM file that was created, the mother-aligned SAM file, and the son-aligned SAM file. These files can be very, very big when you're looking at exome and genome data, so I don't encourage you to open them up to really read them. I encourage you to do computational things to them to extract information out of it. But we're just going to look at the first couple of lines of the father's sample, just so you understand what the SAM file looks like. So I'm going to type in the word head, which will show me the first 5-10 lines. and align SAM. We hit enter. And so here, let me come back up here to this. So this is where I typed in head father, align SAM. So here you can sort of see what genome we're using. If I had all the chromosomes, you'd see them all listed. Here we're only looking at chromosome 20. Here is my read group information that I put in each command. And so that is implemented here. This is the actual command and the version of BWA along with all the options I ran. So it'll always be there inside the inside the SAM file if you ever forgot what command you used to type. And then here's the actual information of the alignments. This is just the beginning line of every FASTQ file for every sequence that has a serial number of the sequencer along with several other things that give the coordinates of where that particular read was in that particular FASTQ file. Here you can see it lined to chromosome 20, that all of it was matched, 151 matches. Here was like 105 matches, 2 insertions, and then 44 matches. You'll have your sequence information, then your quality score, and several cigar strings, which if you want to know more about that, I encourage you to look at the SAM file documentation and to also look up at the cigar strings to understand better what all of that information is telling you. Okay, and that just keeps going. So for every sequence read that was in your FASTQ file, it's represented here, tells you where it was aligned at, what was the position in that particular chromosome region. If it didn't align, there wouldn't be any alignment information here, but the file would contain your unaligned reads as well. Okay, so that's your alignment step. We're done with the alignments. It's now time to go in and mark duplicates. We want to remove any sort of PCR artifacts, things of that nature that would cause GATK downstream to call a SNP falsely. So what we need to do is we first need to sort the file in a coordinate-like order. It's helpful for GATK to have things in coordinate order. Otherwise, it just fails miserably downstream. So at this step, we're going to do this for each of the 3 samples, the father, the mother, and the son. So I'm going to copy this particular sort file. So we're using Picard now, and we're using its tool sort SAM. And I'll explain those parameters that I'm using, but there are plenty more to look at. And so all this is doing is sorting. And so Picard is a jar file, so you need to call Java, and then jar, and then you provide the location of where Picard tools is, and then the Picard jar file. You're using the tool inside Picard called sort SAM. You provide an input, which would be the aligned SAM file that we just created from BWA. And then I'm going to say an output. I want to create a new file called father_aligned_sorted.bam. And then we're giving it the sort order coordinate. Notice that this sort SAM tool turns your SAM file into a BAM file. And these are binary files, so if you were to do a head command on this particular file, it would look like a bunch of garbage running across your screen. It's basically 0-1 binary format, so you won't be able to really read it. If you want to look inside a BAM file, you've got to convert it back to a SAM file. All right, so let's run this on the mother. All right, so that's done. And so now you should see in your directory folder 3 BAM files that have the _sorted.bam. These can now, you can even do it in a SAM file as well, but I would encourage you to take up the BAM file and load those into some sort of genomics viewer to be able to view the reads that are aligning to your genome. So tools like IGV or IGB, or even using the UCSC genomics browser, and I've listed several more here, allow you to view the alignments. It's very helpful to come in and see the actual reads and the different SNPs and indels that were called from your sequence information. All right, so the sorting step is done. Now we can come in and remove duplicates. Now it's in the correct order for GATK downstream. And so now we'll run the mark duplicate step. And again, we're going to use the Picard tool set, and we're going to use the tool inside Picard called mark duplicates. Same command as before just about, except here we're going to call mark duplicates. I'm going to input in the sorted BAM file that was just created, and then we're going to create a new output file that I just called dedupe, short for deduplication. I create a metrics file to be able to see the stats on how many of the reads were duplicated. Gives me an idea if how much PCR artifacts and other things might have caused this duplication to go awry. I want it to create an index so that it can be viewed in IGV better, and I'm telling it that yes, I did sort it. I sorted it in the step before. So copy and paste. Fairly quickly again. Don't expect that to be the case for a whole genome. If you have 100 samples, you'll need to do this for 100 samples. Yes, you can batch job these if you have a computing cluster or just a really high-end computer. You can run this as a batch. You can do for loops and such to run through everything, so you don't have to keep typing these commands out one at a time. This is just a basic introduction to show you the commands that you need to use. So feel free to modify this for your own needs. So we just marked them. We have a dedupe BAM file for each of the samples that was created. One of the steps that's sort of not inside GATK, but I like to look at it occasionally, is how well did my sequence reads align to the genome that I was aligning to. It's very helpful to just sort of get an idea. Was it 90% of the reads? Was it 70% of the reads? Or was it only 10% of the reads? If it was really, really low, 10 to 50% of the reads only aligned to the genome, I would be worried that you have some sort of contamination, whether or not it came from your lab or it came from the sequencing center. You want to get a feel for, well, how many reads that I had actually aligned to the genome. And so a very simple tool to use is inside SAM tools. It's called FlagStat. And it's a very, very simple command to type out. It's so simple that I will actually type it versus copying it. So father dedupe.bam, and it will output a very simple table that will say, hey, there were like close to 52,000 reads total, 99.87% mapped, and that 99.49 were properly paired. Here, if I saw anywhere things, 60% or below, I would be a little worried that I might have something else inside the sample. So definitely take a look at those. Run SAM tools FlagStat. It's just a very quick metric. There are other better tools that look at a whole bunch of other things, but in this example, I just want to know how well do they align. And also, going back to those marked duplicates, you look at that metrics file that was created to get an idea of, well, how many reads were duplicated as well. So you need to look at both of those to get a better understanding of how well my reads aligned. All right, great. We're almost done with that pre-processing phase. You'll start to realize as this recording goes on that this is quite a lot of steps to process, but it is one of the best sets of tools out there to call variants.